Bio 30, welcome to the video on mitosis. We're going to get started. In your notes, you are going to have a section that is called mitosis with a bunch of circles. That is where we're starting. So today's learning intention, I'm learning how cell division affects body cells and sex cells. That's a very generalized idea. Today's success criteria is you can identify which cells, body or sex cells, are going through mitosis. The process is happening during interphase. Being able to diagram or create pictures of prophase, metaphase, anaphase, telophase, and cytokinesis, and identifying some key pieces from each step. So what is mitosis? Mitosis, ladies and gentlemen, is cell division, essentially, in a nutshell. But this is where a parent cell, so an original cell, divides into two identical copies of itself, or what we call two daughter cells. Now, when we go through mitosis, because our body goes through mitosis, it occurs only in what we call somatic cells. Somatic cells are all our body cells. So hair cells, skin cells, eye cells, our eyelashes, our fingernail, mitosis. Okay. The one thing you have to be aware of is mitosis does not occur in sex cells. So sperm and egg, they don't go through mitosis. All right. So if you remember somatic meaning body, that is where mitosis is happening. Now, we are going to talk really quickly about the world of interphase. So interphase, you may recognize it from the cell cycle video. Interphase is pretty much everything in the cell cycle except cell division. So cell division is mitosis. So interphase precedes mitosis. Okay, so everything leading up to cell division is interphase. Interphase has G1, S, and G2 included in it, and we've already talked about those, and we've learned about replication. So when I show you this picture of my cell with my nuclear membrane, my nucleolus, the centriole becomes important later. There's my first set of chromatin. I go through the S phase, and now I have a second set of chromatin. That is the point of S phase, DNA replication. It's going to increase in size, then it's going to go through cell division. So in your notes, I would like you to fill in the part that says interphase. You're going to draw this picture, all right, and label this picture. You do not need to write down everything beside this picture, as we've already talked about what is included in the cell cycle. If you want to, you can, but again, you don't have to. So just pause your video, draw this picture, or watch the whole video and come back to these pictures. This is where mitosis is now, I forgot the words chromatin. So this is now where mitosis starts. We are starting in the world of prophase. So there's quite a few things that happen during prophase. I'm only going to highlight the two key pieces, but I want you to draw the picture. So prophase, step one, here it is. These lovely little things called centrioles migrate to the opposite poles. Okay, so you have two of them, they're going in opposite directions. They have something on them called spindle fibers, which are going to attach to our chromosomes, which you'll see in a minute. But the key pieces I want here are, A, your chromatin condenses into chromosomes, so they become the X. I also want you to write down that the nuclear membrane disappears. Why is that important? Is because we are going to divide the cell into two. If the nuclear membrane was still intact, we wouldn't be able to pull out the DNA from there. So the nuclear membrane has to disappear. And that's what you're seeing here. When it does that, this is the key part that I was telling you about, that the chromatin condenses. So when you're drawing these pictures, it is easier to draw your X's in different colors, because as we move through this and then meiosis, those different colors are going to represent mom and dad. All right, so that might help. If you don't have different colors, that's okay. So this is prophase and what it looks like. There is a step that we are not going to diagram, but I'm going to talk about quickly, which is called late prophase or prometaphase. So it happens between prophase and before we get to metaphase. And what it is essentially the spindle fibers, those centrioles had those spindle fibers, they actually just attach to the chromosomes into the center right on their centromeres. I'm okay with you not understanding prometaphase. It does not matter. As long as you can draw prophase and then you can draw this metaphase, that was what matters. So metaphase, our next step, essentially looks like this. You have the chromosomes lining up at the center. They are attached to the spindle fibers in a line. So we call this sister chromatids aligning at the equator, because remember, in the X form, each one of those arms means a sister chromatid. Now, you do not need to, again, write everything down as long as you write down that the chromosomes line up at the metaphase plate or the equatorial plate, which you can label on your picture right here. That's what's key with metaphase. Okay, so metaphase is all about these spindle fibers are attached, they are lined up, and they are ready to start dividing. So what are we dividing? Well, here is a picture of anaphase. Anaphase is essentially when they break up. So imagine those sister chromatids have a massive sister fight. Me and my sister used to get them all the time. You go to your opposite room, slam your doors, you're just really ticked off, right? It's essentially what's happening here. What is happening is that center centromere, so that dot in the center, is actually being ripped apart, and they are going to now be dragged to opposite sides of the cells. So anaphase looks, anaphase looks like this. Essentially, the spindle fibers have pulled back, and now you have your individual chromosomes attached to the spindle fiber. Remember, they are now single chromosomes because they're not attached together. If they were attached together, they would be sister chromatids. But now that they are pulled apart, they are sister chromosomes. They're just chromosomes. So for anaphase, please draw this. And the only thing you really need to tell me is that they're moving to opposite sides. So if you just put move to opposite sides, chromosomes move to opposite sides, that would be perfect. That hits us into telophase, which is essentially when we get a little bit of division starting between our original cell. So what's happening is now those chromosomes have reached opposite sides. The nuclear envelope is starting to reappear, and those chromosomes are stretching out to go back into chromatin. So for telophase, you're seeing that first initial step of that division of one cell into two. The key parts here, guys, are that we form what we call a cleavage furrow, which is right here, this lovely little dip in, okay? And the other part that you just need to say is that chromatin stretches out and we have a nuclear membrane. So that's kind of right here. Two nuclear membranes, cells lengthen into two cells, okay? So that's telophase. Now telophase naturally rolls into the next one, even though we gave it a different name and it seems like it's its own phase in itself. It's not. Telophase kind of naturally moves into this phase of cytokinesis. So cytokinesis essentially just means now we have two separate cells. That cleavage furrow, that dip in the center, just now fully divides, and we now have two separate cells. We can say that the cleavage furrow will invaginate and create two cells in humans, but in plant cells, it actually turns into the cell wall, which if you remember back to Science 10, is kind of unique to a plant. So if we look at the plant side of it, this is kind of what happens to a plant. I only just want you to draw this for cytokinesis. You can relabel if you want that this is the nucleus with the chromatin in it, but I would refer to these as daughter cells. I would just write yourself a little note saying daughter cells underneath. That is how mitosis occurs. So that is cell division from prophase to cytokinesis. Again, the key parts here, guys, pictures and just two pieces of information for each, nothing crazy, okay? I just want you to be able to identify the picture to the name of the stage and have some key ideas of what's occurring there. If you have any questions, please let me know.